So hello everyone. Today our guest is Dr. Andrew Maltez Thomas from the Computational Metagenomics Lab, CBO, located in Italy. And he'll be presenting his work about cancer and the gut microbiome. So many thanks to you, Dr. Andrew, for agreeing to present your work to us. It's such a pleasure to have you here today with us.
So thank you, Sophia, and thank you, the organizers, for this invitation. And yes, so let me know when I can start.
I think you can start sharing your screen now, okay, since the meeting is already pre-recorded. So thank you.
Okay, can you see the screen?
Yes, thanks.
Okay, great. Right. So, okay, so today I'll be talking a bit about, more generally about how the microbiome and cancer are linked and what are these links, and a bit of the work that we're doing to explore a bit better these links. So yes, I'm Andrew. I'm a postdoc at the Computational Metagenomics Lab, which is led by Nicola Segata at the CBO department at the University of Trento in Italy. So first off, I would like to just talk to you a bit about what is the microbiome. So everybody has this, this concept a bit clearer in their minds before we go forward through the presentation here today. So normally when we talk about the microbiome, we're talking about the microbiota. So this is, these are the microbial members. These can be bacteria, archaea, fungi, protists, algae that are present in a particular microbial community. But the microbiome also encompasses what we also call the theater of activity, which also includes proteins, peptides, lipids, polysaccharides, all of these elements that are produced by the microbiota and are together there in that microbial community. And this also includes other external or internal structural elements that include environmental conditions such as specific pHs, and salinity, together with different microbial metabolites. So like signaling molecules, toxins, as well as other both organic and inorganic molecules. So all of this together is generally what we talk about when we talk about the microbiome. So what we have, what we've seen so far in several studies, landmark studies looking at the human microbiome, is looking specifically, at the microbiota and the different niches of the human body. So we can see that there are body site specific microbial communities. So, the microbes that you find in your oral cavity are going to be different than the microbes that you find in your stool versus the ones that you find in your stomach, whether you have H. pylori or not. And we also saw from these studies that there is a high interpersonal variation. So, the gut microbiome from is going to be very different from different individuals. However, we do see a certain temporal stability within a single individual, meaning that, over time, you tend to, have a more resilient and a more specific gut microbiome or microbiome tailored to your specific body. So, okay, so now I'm going to talk to you. So we know a bit about what the microbiome is, but then there's this new concept that's emerging, which is the oncomicrobiome. So onco comes from cancer, meaning cancer. So it would sort of be what is the cancer-related microbiome. So this, you could define it as different configurations of our microbiome that can influence cancer in several different ways, such as a causal agent in the development and the progression of different types of tumors. And this could be by, some microbial members producing certain bacterial toxins and or metabolites that cause, DNA damage, which I'll talk a bit about more down the line. We can also think about the oncomicrobiome in terms of modulating our response to cancer treatment. And depending on your gut microbiome, you can respond or not to chemotherapy and immunotherapy, depending on what sort of microbes you have in your gut. And also, we can think of the oncomicrobiome in terms of influencing different cancer-associated comorbidities. And these comorbidities can be such as obesity. So we know there's a link there between the microbiome and obesity, and also obesity and cancer. So also cancer cachexia. So these are sort of a bit of the three main areas where we can talk about how the oncomicrobiome exists. So I'll talk a little bit about trying to convince you guys that there is a such thing as an oncomicrobiome. So here we see in this work of 2016, so this is purely epidemiological evidence of the existence of this oncomicrobiome. So here we have in the different columns, different cancer types that were evaluated based on 125,000 cancer cases. And in the rows here, we have the different classes of antibiotics. And you see here that, depending on what was the antibiotic use and depending on the different tumor that the authors looked at, they saw an increased risk for the development of certain types of cancer with the use of antibiotics. And here I'd like to just draw your attention to, for example, lung. We see a pretty consistent signal of antibiotic use and increased risk of lung cancer. We also have here for gastric cancer, prostate cancer, and renal cancer. So again, not only is there an association there between antibiotic use, and the risk of cancer, but also in terms of antibiotic use and the efficacy of immunotherapy treatment. So here we see, I'm showing you an article from 2019, where the authors looked at several different studies, whether these different clinical trials, or whether the patients took antibiotics or not, and whether they responded or not to immunotherapy treatment. So here what we can see is that most of the studies using a meta-analytical approach, what they see is that when you across these different studies, when you took antibiotics use, it decreased the efficacy, the probability of the efficacy of responding to immunotherapy treatment. So these are two strong cases for the epidemiological side of the existence of the oncomicrobiome. Now I'd like to show you a bit more about experimental evidence. So these are all in mice models. So first off, in this paper by Wong and colleagues in 2016, where they had mouse models where they took these mice and they fed them antibiotics, and then they gavaged them with stool samples, either from healthy controls or with stool samples from CRC patients and basically no stool samples at all. And then they did this gavage, and then they sacrificed the animals, and then they looked at different characteristics of these mice. And what they noticed was that mice that were gavaged with stool samples coming from colorectal cancer patients had a higher percentage of polyps compared to those coming from control individuals, as well as those that received no stool samples at all. They also noticed that mice that received stool samples from colorectal cancer patients had a higher grade dysplasia, as you can see here, compared to the other groups, as well as a higher histology score. So here is showing that the stool, let's say the phenotype of cancer, colorectal cancer in this case, could be transferred to mice models. Another compelling evidence for the the oncomicrobiome is in terms of the efficacy of immunotherapy response. So here in this paper by Ruti et al. In Science, what they did was they had their mice models, which they then transferred via gavage to these animals, the stool samples of patients that responded to immunotherapy and patients that didn't respond to immunotherapy. So basically what they see is that if you compare the tumor size of these mice models, you didn't see any difference in terms of the tumor size. So basically the tumors were the same size. However, What changed actually was the tumor size after these mice were treated with immunotherapy agents. So they see that, mice that received stool samples from responder patients actually had a decreased tumor size after the treatment with anti-PD-1 when compared to mice that had received gavage samples from non-responder patients. So now we know what the microbiome is, a bit what about the human microbiome is, a bit about what the oncomicrobiome is. Okay, but how can we study these different microbial communities? Well, I mean, one of the classical ways is, you have your stool sample, or you have your microbial community here. You then proceed to isolate these individual bacteria, and then you do some sort of phenotypic characterization to understand, what bacteria is there, what species, and so on and so forth. But however, if we want to do this in a large scale, I mean, of course, you can also, instead of just doing phenotypic characterization, you can also take these isolates, and then you can then sequence them, and then you can understand a bit more about their genome, what genes are there encoding, and so on and so forth. However, this, if we want to analyze a large volume of both, samples, different patients to get the variability associated with the microbiome, but also you want to look at several different species at the same time, this gets a bit complicated. So this is where metagenomics comes in, which is basically you're just taking whatever DNA is present in your microbial community, and then you're just going to sequence whatever DNA is there. And then through different computational analysis pipelines, and you can then determine who is there, what are the genes that they have, what their abundance is within the community. So it's basically, metagenomics is just a study of all the genetic material within a sample without the need of cultivation. So this is a bit of a workflow of what goes on in the microbiome when you're analyzing it via metagenomics. So basically you have your microbiome sample where you've just sampled your microbial community directly from the environment. This can be from, the human guts through stool samples, ocean water, soil, whatever. And then you're gonna collect the sample. You're gonna have your microbial community where you're gonna extract the DNA. So you have all the DNA there. But it's also important to note that when you collect these in microbial communities, it's important to see and annotate different metadata information, demographics, information on the environment or the host, latitude, longitude, whatever sort of information you can jot down at the time, because this will be really important for you to understand a bit more about what's going on in your microbial communities. Right, so then once you have those isolated DNA, you then proceed to DNA sequencing, where you will have what we call your metagenome, which is in normally FASTQ files, with a bunch of sequences. So then you have what you call your metagenomic data set, where you have these different metagenomes coming from different microbial communities, and these can be in the size of about terabytes. So then when you have these, you then have to proceed to do some quality control of these metagenomic data sets, where you have to sort of remove low quality sequences, remove host DNA contamination, if there is any. So for example, stool samples that we work with, we routinely remove, whatever human DNA contamination is there. So we know from previous work, that stool samples have a relatively small amount of human DNA present in there. So it's not, we don't lose that much information. So then once you have these pre-processed metagenomics data sets, again, in size of terabytes, you then proceed to use these sequences together with reference information, such as reference genomes, annotated protein families, annotated pathways. And this is where you really do the sequence analysis, where the end goal is basically for you to do, you have two different routes here. Once you have these pre-processed metagenomic data sets, you can do what we call assembly-based profiling, which is, basically just overlapping reads to create larger reads or contigs, and then you can use these to understand a bit more what genes, species, and functions and pathways are present within your samples. And there's also the read-based analysis, where you don't really try to do any assembly, you just overlap these reads and then proceed to taxonomic and profiling. And then after you have this sort of information, the contigs, the genes, what species, the functions and the pathways, then you can lead to, together with your metadata information, you can look at, some sort of biomarker discovery. You can do sort of classification, such as supervised machine learning, get cluster, phylogeny reconstruction, strain tracking. There's a bunch of different stuff that you can do with this data. So it's a very rich data set. And then, of course, you have to do the validation with, an independent cohorts, just something that we strongly believe. So as you can see, this is a very multidisciplinary approach that involves several different, expertises, from computer scientists to statisticians, microbiologists, microbial ecologists, and biotechnologists. So, okay, now I'm going to talk a bit specifically about, what we know so far and what is being hypothesized in terms of, mechanisms of how, the gut microbiome can lead to carcinogenesis in colorectal cancer. So carcinogenesis is basically, the malignant transformation of normal epithelial cells. So there's three main hypotheses that this can occur through basically changes in your microbiome. So where you have your gut microbial communities here, something leads to a dysbiosis. So basically, there's an alteration of, the steady state of your gut microbiome. And then this can lead to the translocation of some certain bacterial species that can translocate from the normal epithelium tissue to sort of get some microbial antigen proteins that can then lead to several different pathways associated with the immune system that can ultimately lead to sustained inflammation and proliferation of cells and develop into malignant cells. There's also through the production of genotoxins, so colobactin or CDT producers. So I'll talk a bit more about these in the coming slides. But the idea is that certain bacterial members can produce some certain toxins that are genotoxic, so they cause damage to DNA, and then this damaged DNA can then lead to the malignant transformation of the normal epithelium cells. And also through the production of different microbial metabolites. So for example, there's the microbial conversion of ethanol into acetaldehyde. And acetaldehyde is extremely carcinogenic, and this can then also lead to DNA damages and can then lead also to the development of cancer. There's also bile acid metabolism, and that can also cause genotoxic effects. So I'll talk to you a little bit about one of the first identified, one of the strongest cases where we see a causal relationship between a particular bacterial species and the development of colorectal cancer, which is Fusobacterium nucleatum. So this is a species that is commonly found in the oral cavity, more commonly found in periodontitis, and it's rarely found in the human gut microbiome. It has been shown to potentiate tumor cell growth. So when this species was infected in colorectal cancer cell lines and fed to mice and in xenograft models, they all led to the development of tumors. And the mechanisms by which this particular bacterial species can cause tumor development, it goes from enhanced tumor cell adhesion to invasion. It also helps modulate the host immune response, and it's prevalent in about 30% of colorectal cancer patients. But I mean, we still don't really know how this bacteria, which is commonly found in the oral cavity, reaches our gut. I mean, we would assume, there would be some sort of mechanism that prevents all species from reaching our gut, but this actually seems to be quite common. So I show you here some results from Schmidt, paper by Schmidt et al. And eLife 2019, where they looked at what we call the oral-fecal transmission in healthy individuals. So basically, in this article, they're looking at whether they could identify species that were common inhabitants of the oral cavity inside the gut of the same individuals. And basically using about 400 healthy individuals and looking at about 300 10 species. They say that about 125 of these species, 77% of them show really, prevalent oral fecal transmission. So basically, this means that species from the genus Streptococcus, Veillonella, Actinomyces, and Haemophilus were commonly found in both the oral cavity and in the gut of the same individuals. So this seems to be something that is not so uncommon among healthy individuals, where you have species from your mouth reaching your gut. So they also looked at this oral fecal transmission in different diseases, including colorectal cancer, which is something that we also looked at, sort of at the same time in our data sets. So here, what I show you here is, we have the number of oral species that we found in our data set of controls and carcinoma colorectal cancer patients. So basically here we have the different data sets here, and here we have the coefficient estimates. So basically, when this coefficient estimate is to the right, that means that there's a greater oral species richness in carcinoma patients. And when it's to the left, that means to controls. So basically you see that for most of the data sets, we see a increased oral species richness in carcinoma patients. This is something that they also saw here together in colorectal cancer patients that had an enrichment oral species, and also they also saw the same thing with rheumatoid arthritis. So this seems to be something that's pretty common in colorectal cancer. So in this paper of 2016, the authors proposed sort of a mechanism of how everything is linked: oral species, biofilms, and also colorectal cancer, where you have species such as Fusobacterium, nucleatum, poriformonas, Streptococcus, and parvimonas. These all somehow reach the gut. They together they form these biofilms that can then lead to low-grade sepsis and constant inflammation, which can then lead to the malignant transformation of these epithelial cells. So just a side note here, maybe it's important for us to consider the oral microbiome for different diseases. As this review last year that came out last year pointed at several different disease links associated with oral species and the oral microbiome. This can go from diabetes, from rheumatoid arthritis, cystic fibrosis. So that's maybe something interesting to look at. Okay, so the next species that I wanted to talk to you a little bit about is Escherichia coli. So basically, this is a species that is a commensal resident of the gut microbiome, and it's about prevalent in 60% of healthy human feces. It's been shown to promote homeostasis of gut microbial communities in the luminal tract. However, some strains have been shown to carry a combination of virulence genes that enable them to cause intestinal and extra-intestinal infections. And through phylogenetic analysis, we know that there's four main phylogenetic groups: A, B1, B2, and D. And these pathogenic strains that carry these virulence genes belong mainly to groups B, 2, and D. However, most of the fecal strains that we find in healthy human fecal samples belong to groups A and B1. And there are E. coli from the B2 group that can carry what is called a polypeptide non-ribosomal peptide synthase operon, so PKS for short, that's responsible for the production of the genotoxin called colobactin. So I think I mentioned this a bit in the start of the talk. And yeah, so here, this PKS positive E. coli, so previous studies have been shown that they're prevalent in about 20% of healthy individuals, in about 40% of individuals with irritable bowel syndrome, and in about 60% of patients with colorectal cancer. So in this work published in 2019 by Weirgo colleagues, they looked at the prevalence, the abundance of PKS, the PKS colobactin operon in colorectal cancer patients compared to controls, and they found a significant increase in colorectal cancer patients. and also a year later published in Nature, where the authors looked at whether they cultured PKS positive or PKS negative cultures. They saw that cell cultures. They saw that PKS positive cells that were cultured with these PKS positive had a different mutational signature compared to cells that were incubated with an inactive form of Caulobacterium, basically. So this led to a higher number of indels and a higher number of also single base substitutions. And so I think this shows the importance of when we're looking at the gut microbiome, the importance of looking at the differences between strains, because here we can see clearly that whether E. coli has PKS or does not have PKS seems to have a pretty big impact, both in the development of tumors and also with the mutational signature of the tumors that had PKS positive strains. Another microbe that I would quickly like to touch on is Bacteroides fragilis. So this is another species that is highly prevalent in the human gut of healthy individuals. However, it has been shown that it can also promote carcinogenesis via the induction of pro-inflammatory toxins. And one of these toxins is the enterotoxin BFT, so Bacteroides fragilis toxin. And this is generally called ETBF, so it's the enterotoxigenic Bacteroides fragilis. And this work by Wu and colleagues in 2009 showed that whether if you had Bacteroides fragilis, whether it's the enterotoxigenic or not, it really increased the number of tumors in their mice models. Another interesting microbe that has been shown to have some evidence to have carcinogenic effects is Streptococcus gallolyticus. So this is an opportunistic pathogen that causes bacteremia and infective endocarditis. There's been a strong clinical association with CRC in that about 60% of patients that were found to have Streptococcus gallolyticus bacteremia also were found to have colon adenomas and adenocarcinomas. And yeah, in this work published in 2017, the authors showed that mice that were administered with Streptococcus gallolyticus had significantly more tumors, a higher tumor burden, and higher dysplasia grade compared to mice that were, even administered saline solution or that were administered Lactococcus lactis as a control. And then I think lastly, I'd like to talk a bit about Campylobacter jejuni also. So Campylobacter are not very prevalent in healthy individuals, being found in about 5% of healthy individuals. However, Campylobacter jejuni is an opportunistic pathogen in the human gut. And some of these Campylobacter jejuni strains also carry the bacterial genotoxin cytolithodistentin toxin, so the CDT, which can also cause host DNA damage. So this is showing work from he et al. In Guts 2019, showing that again mice that were administered with these strains of Campylobacter jejuni that had CDT caused increased number of tumors in their colon, and also you can see their colon is very different than those control mice. And then I'd like to talk a bit about, so not only certain bacterial species have been associated with colorectal cancer, but also different microbial metabolites. So here I term them oncometabolites. So these are metabolites that have been shown to have some sort of oncogenic or carcinogenic potential. So one of them here that talk about is the secondary bile acids, where you have a high fat diet. This high fat diet leads to an increase in primary conjugated bile acids, and these are then degraded by bile salt hydrolases into unconjugated bile acids. And then through this multi-step 7-alpha dehydroxylation, we get to what was called secondary bile acids, such as decoxylic acid. And these have been shown to have tumor promoting activity. And we know that, certain members of the gut microbiome have more 7-alpha dehydroxylating genes. So part of the dehydroxylation pathway, and these include, Clostridium sitidens and Clostridium immunae, and many different other clostridial species. So in work by, again, Weirbo in Nature Medicine 2019, the authors again compared, colorectal cancer patients to control individuals, and what they found was an enrichment of secondary bile acid genes in colorectal cancer patients. So here are, part of the bioperon of these 7-alpha dehydroxylation genes. And you can see here the different cohorts, and there was an increase compared to controls of these genes in CRC patients. And this was also confirmed by qPCR. So another oncometabolite that I would like to talk about also is trimethylamine. So trimethylamine is basically it's an amine that's comes from mostly our diet. So we have here, for example, choline and carnitine, which are present in our diet. These can be metabolized into trimethylamine. Trimethylamine is then oxidized in our liver by human enzymes that oxidize TMA, which is then becomes TMAO. So oxidized trimethylamine, and this then can go into our circulation, and has been TMAO has been associated with foam cell formation, platelet hyperactivity, atherosclerosis, thrombosis, several inflammation markers, stroke, heart failure, and chronic kidney disease. So basically, how TMA is broken down to is through some of microbial enzymes present mostly in the large bowel. So carnitine and both choline can be broken down into TMA. So here we have choline as an example. So we have these enzymes that can degrade carnitine and choline into TMA, which then is oxidized into TMAO. So I'd like to draw your attention specifically to these two enzymes here, which is CUT-C and CUT-D. So CUT-C is the choline TMA lyase activity. So this guy is the main responsible guy of breaking down choline into trimethylamine. Then we also have this CUT-D, which is the activating enzyme of CUT-C. So we were interested in looking at these particular genes in regards to colorectal cancer patients. So we looked at the abundance of CUT-C of this TMA producing enzymes in colorectal cancer patients, and what we found actually was that there was an increase of these enzymes in 4 of the different data sets that we looked at. So here are the different data sets that we looked at here. This is the abundance in controls, adenomas, and carcinoma samples. And this increase was also significant in meta-analysis. And also when we confirmed this increase also by a different approach, which here basically we're doing metagenomics, but we also confirmed this increase in by qPCR. So we also found that these enzymes, not only were they more abundant in colorectal cancer patients, but it also seems that colorectal cancer patients have different types of sequences coming from different microbes. So basically here we have a phylogenetic tree that was built with the sequences, the CUT-C sequences coming from the different samples from the metagenomes. And what we can see here in terms of, this is the odds ratio. So how likely, our control samples, how likely are they to have a CUT-C coming from an uncultured bacterium? So basically they're very likely, whereas colorectal cancer patients had an odds ratio of having basically CUT-C sequences coming from E. coli, Clostridium species, Hungatella, and this was something that also we thought was interesting. So not only was there an increased abundance, but also where these CUT-C sequences were coming from, what microbes also seem to be different between controls and colorectal cancer patients. So as I mentioned a bit at the start of the talk, there's different ways that the gut microbiome can be associated to cancer. And this is another one of the ways, which is it can modulate the response to cancer therapy. So this is, for example, a paper published by Geller in Science 2016, where they found that different taxa within the pancreatic tissue could express an isoform of cytidine deaminase that could then inactivate gemcitabine, which is like a chemotherapeutic drug that's used to treat pancreatic cancer patients. And basically, some of these species could then lower the local concentrations and then, confer resistance to these patients to this particular drug. And basically here, what they show here is, along the time, how much of this particular chemotherapeutic drug was remained. So basically, we had, if you look at, there were some species such as this Acromycobacter and this Escherichia coli strain, that the amount of this chemotherapeutic drug that remained decreased a lot, whereas some other species actually maintained the percentage of this drug. And then here they also show that antibiotic-treated mice respond to this particular chemotherapeutic drug. However, control-treated mice displayed rapid tumor progression. So again, showing here that there is something going on here in terms of the microbiome and response to this particular chemotherapeutic drug. So also I mentioned that the gut microbiome can be also associated with other cancer comorbidities. And in this paper published in Nature, they show like how a microbial metabolite can induce obesity-associated hepatocellular carcinoma. Again, these are using mice models. So basically, they show that this gut bacterial metabolite, the deoxycholic acid, provokes senescent cells to develop a secretory profile that's composed mainly of inflammatory cytokines, chemokines, and proteases. And this particular phenotype then facilitates the development of hepatocellular carcinoma. And then basically, they show that mice that were fed a high-fat diet had more Clostridium species, and together with other bacilli. We can also think of applications of the microbiome. So what other applications can the microbiome have in terms of cancer? So first off is using the microbiome to diagnose cancer. So these are some of the papers that have come out in the past few years that have been shown the potential of the microbiome to diagnose cancer, both in terms of hepatocellular carcinoma and also from blood and different tissues. So I'll talk a bit more in depth about what we did. So basically, we looked at whether the gut microbiome could be used to detect colorectal cancer across the different public data sets that we have. So here's basically a supervised machine learning approach where these are the different cohorts that we used. And then here is basically what we call the cross prediction matrix. So the values that you see here are basically AUC used when we use the cohort FANG, when we train our supervised machine learning algorithm on FANG and then test on FANG. So basically this is a cross-validation strategy. So basically when we trained and tested on the same cohort, we saw that some data sets were able to predict colorectal cancer better than others. This is just based solely on the gut microbiome. However, when we used the approach where we trained on one data set and then tested on the other, we saw that there was a poor transportability of the information that was learned from one data set to the other. So what we did was try to use an approach where we combined the different data sets. So basically here we show the different AUCs in the different number of training sets that were used to train the supervised machine learning classifier. And what we can see here is that as we increase the number of training data sets, the AUC values increase. And it was so much so that we were able to reach an average AUC of 0.84 when we combined different data sets and then trained and then tested on the left out data set. So this is what we call the leave one study out strategy. or loso. So this was nice because, at the same time, Whitwer et al. Were doing something sort of similar, and they had a sort of partial overlap in the samples. However, they were using a different taxonomic profiling approach and different machine learning models. In this case, they were using lasso, whereas we were using random forest. And yeah, and what they see here is also the same thing for the different data sets. If you use the leave one study out validation, you can actually increase your detection of colorectal cancer to the point of reaching values of an average of 0.84. So another application of the microbiome is by modulating the microbiome in colorectal cancer patients, in cancer patients, through what we call the FMT, so through fecal microbiota transplantation. So in this study published this year in Science, the authors gave patients that didn't respond to immunotherapy with anti-PD-1 therapy in melanoma patients. So basically, they gave these patients FMT from 2 donors, and then they looked at whether they responded or not. And what was interestingly is that the patients that previously hadn't responded, 40% of these actually started to respond after being, after receiving the FMT. And these patients that responded exhibited increased CD8 T cell activation, decreased frequency of interleukin expressing myeloid cells. And here, as you can see, these are the different patients that they evaluated. And here is the change in the tumor volume compared to the start of the treatment. So you can see that there are some patients that, even after FMT, didn't respond to immunotherapy. However, there are 6 patients here that they actually responded to immunotherapy just by changing or receiving a transplant from a more healthy fecal sample. And this is other applications of FMT. So this is another application here of FMT where the authors here looked at transplanting, doing FMT to patients so that they would stop having diarrhea. So a big problem of tyrosine kinase inhibitors, so TKI inhibitors for renal cell carcinoma, is that a lot of the patients have very severe diarrhea, and it's so much so that sometimes they have to stop the treatment just because of how severe the diarrhea is. So in this work, what the authors did was basically give FMT. So they have 2 groups. We have a group that received the FMT from 2 different donors, and they had a group where they didn't receive FMT. So basically, it was a placebo. So there was no real FMT going on. And then they compared, the diarrhea across these 2 groups. And what they found was, that FMT group actually had a full resolution of the diarrhea after 4 weeks of treatment in about 70% of patients, whereas in the placebo group they saw no patients that had their diarrhea resolved. And this study was supported also by showing that there was a clinical success. So basically, we could see that there was a strong engraftment of donor both species and strains. So here you can see in these graphs here, for example, that before FMT there was no difference between how many shared strains you had with yourself before receiving FMT versus how many shared strains you had with the donor recipient. And basically we see that that there was a significant fraction of shared strains with donors even 4 weeks after treatment with the FMT. And most of the species that were transferred from the donor to the patient were basically Bacteroides species, but also here Akkermansia mucinifera seem to be very commonly transferred from the donor to the recipient. So with that, I hope I have given you guys a brief overview of the microbiome and cancer. So basically how the microbiome can lead to tumor development and or progression through certain specific microbes and or metabolites, how the microbiome can be associated with different cancer therapies, and how the microbiome can be associated with different cancer-associated comorbidities. And also some promising applications of using the microbiome both in the treatment and the diagnosis of treatment of different cancers. So with that, I'd like to thank everybody at the Computational Metagenomics Lab, the PI Nicola Segata, and especially Paulo Munghi, which a lot of the work that I showed here was done in collaboration with him, and and all of our collaborators as well as our funding institutions. And yeah, I'll be happy to take questions. Thank you.
Thank you, Dr. Andrew, for your great presentation. I really appreciate the efforts you put into it. I have a first question, if I may ask first. So you talked about Escherichia coli. Do you think that antibiotic therapy or the intake of antibiotics may influence the abundancy plots in the during the metagenomic analysis?
Yes, for sure. So I think antibiotic use is something that people are giving a lot of attention to right now, and I think we need to understand better what are the actual effects, because we see this both in terms of epidemiological evidence of antibiotic use and the increased risk of cancer, as well as the decreased efficacy of cancer treatments. So I think, yes, especially in the case of E. coli, to better understand, if by taking antibiotics, do you kill the E. coli, the good E. coli, and open up space so that the PKS positive E. coli can colonize. So yeah, I think antibiotics is something that is going to be a very hot topic in terms of the gut microbiome in the near future. So I think recording in progress.
Sorry about that. So I think we have some questions in the chat box. So Dua is asking, what tools do you recommend for most for the 16S RNA analysis?
Right. So I think regarding the 16S analysis, I think one of the major things that you have to think about is let's say what level you're going to be looking at. So we have what we call the OTUs, so operational taxonomic units, versus what is now what we're calling ASVs. So basically the amplicon sequence variants. So I think this is the first major decision, and the tools that you use for one or the other will change. But I think making sure that it makes sense to you, what sort of information makes sense to you is good. So I personally really like Data2. I've used Data2 for amplicon sequence variants, and I've also used a lot the uparse from Robert Edgar to create operational taxonomic units. So those are the two tools, let's say, for sequence clustering that I think are really nice. Also Mother, there's so many tools. I didn't talk about particular specific tools here in this talk, but yeah, I'd be more than welcome to share some other slides I have regarding specifically microbiome, the 16S metagenomics shotgun analysis. So.
Thank you for your answer. It was very insightful. So the second question is, do you have any idea about the success rates of fecal transplantation?
Right. So yes, so that's another hot topic right now in the area. So I think the field is really trying to understand what makes a successful FMT. Is it the donor? Is it the recipient? There are questions addressing whether we should treat the recipient with antibiotics prior to them receiving FMT. So I think these are all questions that we need to look at, and there's some articles starting to come out to try to address these questions. Is it a donor thing? Is it a recipient? Do we have to use antibiotics? So yeah, I think that's a complex question with complex answers, but I think the field is starting to move towards understanding a bit more what makes FMT successful or not.
All right, thank you. So one more question, I guess. What are the challenges you confronted during the metagenomic analysis?
Well, I think one of the challenges that you have to keep in mind is that you're dealing with a large volume of data, and basically you're crunching down terabytes of data to like make a a taxonomic abundance file that you're then going to do some statistical analysis with. So yeah, and so I think as as the field progresses, this will become larger and larger. So you'll be dealing with we were dealing with, I think, 900 samples, but now, we're going to start seeing, studies that are, 20,000 samples, 30,000 samples. And this becomes, whatever you're going to do, you have to be able to do it at scale. So whatever you could do before with 900 samples, you have to be able to do it efficiently at 9,000 samples, or, so yeah, I think basically scalability, I think is one of the things that, yeah, I would suggest people think about doing everything efficiently and fast at scale.
I think this must be really challenging in regard to the budget sometimes, like
yes,
having
also
10,000 sample. Yeah.
Yeah, well, I mean, here's the thing. You have to consider that there's a lot of samples already out there. So you could always use publicly available samples together with, so this is a strategy that we try to use a lot. So I didn't mention in my talk, but we have what we call the curated metagenomic data resource. So this is an R package that collects, that has the taxonomic abundances and functional potential abundances of, I think, I don't know how many samples there are now, but there's going to be a version 3 with like 30,000 samples, and these are all from publicly available data sets. So, I mean, that's another, if you don't have the budget to, analyze a large volume of samples, there are resources out there that have collected publicly available data sets, both metadata, and done all the work and made this available to the community. So yeah, this is always a good way for you to, also validate your findings, whatever you find in terms of your cohorts or your samples, trying to look at whether you can reproduce those results in publicly available data sets as well.
All right, sounds great. Thank you so much for answering our questions. So if there isn't any other questions, I think we can wrap up the session. So thanks again, Dr. Andrew. It really is a pleasure to have you today with us. Thank you.
Thank you, Sophia, and thank you to all organizers and all people watching and that sent in questions. Thank you.